Hello, Acho. Let's see the question. So the question says, which of the following restriction endonuclease enzyme produces blunt end in DNA? So इन चारों ऑप्शन में से हमें आइडेंटिफाई करना है कि कौन सा एंजाइम blunt ends produce कर रहा है DNA में. So the key concept in this question is to first understand what are restriction endonucleases. So this is a fairly easy question. So restriction endonucleases are actually enzymes that can cleave DNA at specific sites only. So they are nucleases, that is, they can cleave nucleic acids, but since they are restriction endonucleases, therefore they cleave DNA at specific sites. So restriction endonucleases recognizes a sequence of nucleotides known as the recognition site, and this recognition site, once recognized by the restriction endonuclease, then it can cleave the DNA into two fragments. Now, the cleaving of the DNA into two fragments can result in the formation of either a blunt end or a sticky end at the end of the cleaved DNA fragments. So now if we see the options, so the first one is BAM-H1. So BAM-H1 is a type of restriction endonuclease, and the DNA sequence which is recognized, or the recognition site which is recognized by the BAM-H1 is as follows. That is G, G, A, T, T, C. So this is the sequence, the recognition site for BAM-H1. Now, this BAM-H1 cleaves the recognition site at this point and this point. So now, due to the cleavage, it results in the formation of two DNA fragments as follows. So one of the DNA fragments will have the following end, and the other DNA fragment will have the following end. So now we can see that there are two flanking ends in the two DNA fragments present. So this flanking end of one DNA fragment and the other flanking end of the other DNA fragment is known as the sticky ends. So this is one sticky end of the one DNA fragment, and this is the other sticky end of the other DNA fragment. So we can say that the cleavage with the help of BAM-H1 results in the formation of two DNA fragments with sticky ends. So it does not give a blunt end, but it gives a sticky end. Now similarly, for the second option, that is EcoR1. Now this enzyme also has the recognition site as follows: GAATTC, and the complementary sequence as CTTAAG. So this is the recognition site for EcoR1, and the cleavage with EcoR1 takes place at this point and this point. So therefore, the two fragments of DNA which will be formed due to EcoR1 will be as follows. So this is the two DNA fragments formed. Now again we can see that we have sticky ends here as well. So this is one sticky end and this is the other sticky end. So therefore again echo R1 results in the formation of sticky ends. So echo R1 also cleaves the DNA in such a way that it results in the formation of two DNA fragments with sticky ends. Now, if we see the third option, so the third option is Hae-III. So Hae-III has the recognition site as GGCC and the complementary sequence CCGG. Now Hae-III cleaves the DNA at this point and this point. Now, if we see the two DNA fragments formed due to the cleavage by Hae-III, then we see that there are no flanking ends which are formed. So we can say that there are no sticky ends, but they are blunt ends. So due to the cleavage by Hae-III, the two DNA fragments have blunt ends because there are no flanking nucleotides present in the two fragments. So now if we come back to the question, so the first option was BAM-H1. So this was giving sticky end and not blunt ends. Therefore, this won't be our correct answer. Then second option, EcoR1, was also giving us sticky ends and not blunt ends. So again, this won't be the answer. The third option says Hae-III. So Hae-III was giving us blunt ends. So this will be the correct answer. So the final answer for our question is option 3, that is Hae-III. Hope you understood this well. Best of luck. Thank you.